Hello, everyone, and thank you for joining us for the Cancer Research Institute's new webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Tuesday, June 8th, 2013. My name is Brian Brewer, and I'm the Director of Marketing and Communications at the Cancer Research Institute. Cancer Research Institute is a nonprofit organization established in 1953 to support scientific research aimed at harnessing our own immune system's power to fight cancer. In light of exciting successes we are now seeing with cancer immunotherapy, the Cancer Research Institute has declared June Cancer Immunotherapy Awareness Month. We present this webinar series, 5 in total, as a way to introduce you to some of the world's top scientists who are carrying out the important research that is leading to cures. Now, over the next 40 to 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert about how this new treatment modality is overturning what we think of cancer or how to treat it. After a 15 to 20 minute presentation from our speaker, we will open up the questions to you that you can submit by filling out the Q&A form at the bottom of your screen, the lower right side. As you submit those questions, I'll field them and forward them to Dr. Wolchok, and hopefully we can get to answering as many questions as possible during the webinar. So before I introduce Dr. Wolchok formally, I'd first like to thank our sponsor, Dendreon Corporation, whose generous support has made this webinar series possible. Now it's my pleasure to welcome our presenter today. Dr. Jedd Wolchok is Director of Immunotherapy Clinical Trials at Memorial Sloan Kettering Cancer Center in New York City, where he is also an Associate Attending Physician in Melanoma Sarcoma, is head of the Swim Across America Laboratory, and is an Associate Director of the Ludwig Center for Cancer Immunotherapy. He is also the Associate Chair of Medicine and leads the Immunotherapeutics Clinical Core at Memorial. Dr. Wolchok is also an Associate Director of the Cancer Research Institute Scientific Advisory Council and Director of the Cancer Vaccine Collaborative, or CVC. The CVC is a joint initiative between the Cancer Research Institute and the Ludwig Institute for Cancer Research, focused on developing safe and effective therapeutic vaccines and other immunotherapies for many different types of cancers. Dr. Wolchok is a real leader in the field of cancer immunotherapy, as can be attested most recently by the extensive news coverage of his work in the New York Times, the Wall Street Journal, NBC Nightly News, CNN, and other major news outlets. He's spearheaded clinical trials of immunotherapies that have saved lives, and in some patients has reversed very advanced cancers, sometimes to the point of complete remission. So we're looking forward to hearing more from Dr. Wolchok about how these therapies are saving patients' lives. His work is a strong example of how immunotherapy is transforming cancer treatment. Dr. Wolchok, we're delighted that you could take time out of your busy schedule to be with us today. And with that, I'd like to welcome you. Thank you for joining us, Dr. Wolchok.
Oh, thank you very much, Brian. It's a pleasure to be here and to help the Cancer Research Institute launch this inaugural Cancer Immunotherapy Awareness Month. It's really a very important step forward for the field, and I'm just so pleased to be part of it. I'm going to speak today about sort of my views of cancer immunotherapy, why it's important, what history has taught us about how to use the immune system to treat cancer, and then more recently what the most current results are in our attempts to optimally use the immune system as a way to treat cancer. Just as a brief historical synopsis, there has been over a century of interest in how to use the immune system to effectively treat cancer. And these observations actually stem from some very early data that were collected by Dr. William Coley, who is actually the founder of the Cancer Research Institute. Dr. Coley was the head of surgery at the hospital that now is Memorial Sloan Kettering, and he made the very insightful observation that patients who developed wound infections after surgery for cancer had better clinical outcomes than patients who did not have infections after surgery for cancer. This was a very insightful hypothesis that he made, in fact, that the presence of an infection could lead to stimulation of the immune system, and that the immune system could, in fact, be part of the control of cancer. Remember that these observations were made in the late 1800s when there really was no knowledge of, at any kind of molecular or cellular level, what the immune system was all about. And so that made his thoughts even more prescient at the time. Coley actually went on to develop a therapy called Coley's toxins, which was a mixture of heat-killed bacteria that were injected into tumors. And by an accounting done by his daughter, Helen Coley Nautz, up to 30% of patients who had Coley's toxins seemed to benefit from it. However, starting in the 1930s, interest waned in cancer immunotherapy because of the rapid emergence of other effective therapies for cancer, including radiation therapy in the 30s, and then in the 50s and 60s, the emergence of effective chemotherapies. In the late 80s, early 90s, basic science, much of which was funded and supported by the Cancer Research Institute, led to a much deeper understanding of what was at a molecular level happening in the immune system, and to the development of more effective immunotherapies. And I think really the first step forward for the whole field of cancer immunotherapy, which seems so logical, that the immune system can so adequately control infectious diseases and can be so potently stimulated to prevent illness with vaccines for infectious diseases. Why could the immune system not be so well directed toward targets on cancer until really very recently? The first step forward, I believe, really took place in the mid-1980s, early 1990s, where we began to learn what substances and what molecules turned the immune system on and off. And one of the first of these molecules is one called interleukin-2, or IL-2. And IL-2 was actually first identified as a growth and differentiating factor for a type of immune cell called the T cell. And T cells are one of the ways in which the immune system can try to control infectious diseases and cancer. And interleukin-2 is a very potent way to stimulate the growth of these cells. And in studies that were first done at the National Cancer Institute by the surgery branch, Dr. Steve Rosenberg and his colleagues, high doses of interleukin-2 were administered to patients with a variety of cancers, and two specific kinds of diagnosis were associated with response to interleukin-2, and those were metastatic melanoma and metastatic kidney cancer. And to this day, there are a significant number of patients who have actually been cured with the use of high-dose IL-2 for these diagnoses. In the case of melanoma, it's probably between 3 and 5% of patients who seem to be cured. And I'm talking about remissions that last over 10 years, sometimes 15 or 20 years. And to me, this is a very important fact because it indicates that the immune system, when properly tuned, can control cancer in a very durable way. Because interleukin-2 does not affect the cancer cell directly. It actually is, as I said, a growth factor for T cells, and it's the T cells that go out and do the hard work of killing the cancer. So with that in mind, IL-2 was explored in a variety of cancers. It's become an approved therapy in many countries for treatment of kidney cancer and melanoma. But the truth is that it does not benefit as many patients as we would want it to benefit, and it does have some very significant side effects. And so for the next few years, between the late 80s and mid-90s, additional preclinical efforts were directed at trying to understand further what the immune system did in terms of controlling cancer, and specifically what other molecules could be either stimulated or inhibited in an effort to try to augment the immune response to cancer. And again, here is where investigators from the Cancer Research Institute partnered with the Ludwig Institute for Cancer Research, were able to learn a lot about vaccines for cancer through our collaborative network, which Brian mentioned, the Cancer Vaccine Collaborative, to try and develop more effective ways to stimulate the immune system to recognize specific targets on cancer. And we learned several things, really. We learned that there are a family of proteins on cancer cells that can be very rational targets for the immune system to use as vaccines. These are called cancer testis antigens, and because they're present only on normal germ cells and on cancers. And we have developed a very robust vaccine program looking at this in a variety of malignancies. But we know that responses to vaccines alone have in general been disappointing in terms of their ability to stimulate a truly sterilizing or protective response against cancer. We need to overcome more of the immune suppressive influences that cancers throw up to try to thwart the immune system. Cancers have survived and evolved, in fact, to really grow in the presence of significant efforts that the immune system presents to try to control them. But the cancers actually have devised a number of different ways to try to evade the patient's immune system. And only recently have we begun to understand this in a very detailed way that has allowed for the development of therapies. So one of these ways that the immune system is actually dampened is through a series of inhibitory checkpoints or molecular breaks that normally prevent the immune system from achieving a state of full and persistent activation. And one of these molecular breaks is a molecule called CTLA4, and it is present on activated T cells as a normal way to try to prevent T cells from achieving a hyperactivated state. We know that it's important for there to be these checks and balances in the immune system, because if the immune system were left only with a mechanism of getting activated and no way of being inhibited, then patients could end up with more common autoimmune disease, and T cells could even die from hyperactivation. So what we have learned is that there are ways that you can temporarily inhibit these immune molecular breaks with medicines to try to cause the immune system to be activated for short periods of time, so they might recognize cancer. And in the case of this molecule, CTLA4, the lead scientist for the Cancer Research Institute, the scientific director named Dr. Jim Allison, who now leads the immunotherapy program at the MD Anderson Cancer Center, made some very striking and insightful insights. He hypothesized that if one could block one of these molecular breaks with a protein and an antibody, that you could allow the immune system to become more activated than it otherwise would be in mouse models. And based upon his very important observations in preclinical studies, various pharmaceutical companies had developed medicines that block this inhibitory molecule called CTLA4, this molecular break, in humans. And one of these medicines is called ipilimumab, and it actually is an antibody that blocks this molecular break. It blocks CTLA4 in human T cells, and therefore allows these T cells to become more activated than they otherwise would. And this medicine was evaluated in a series of clinical trials in patients with several cancers, including melanoma, and kidney cancer, and lung cancer, and prostate cancer, as well as smaller numbers of patients with other cancers, and was actually approved by the U.S. Food and Drug Administration in 2011, because it was the first medicine ever to improve the overall survival of patients with metastatic melanoma. And this was really a landmark for several reasons. First of all, metastatic melanoma to that point in time was a disease that desperately needed new avenues of therapy. And it was a disease where the life expectancy for a patient with advanced melanoma was less than a year. And we really needed new medicines to go beyond the chemotherapy, which was approved, and the high-dose IL-2, which, while it benefited some patients in a very meaningful way, it benefited too few patients. And so the approval of ipilimumab to treat melanoma was an important step forward for the community of patients with metastatic melanoma and their caregivers, but it was also a landmark for the field of immunotherapy because it put a new treatment on the map that extended overall survival in a rigorously controlled clinical trial. And so ipilimumab now is in common commercial use in the US and other countries around the world for patients with metastatic melanoma. It benefits somewhere just above 20% of patients with melanoma by extending survival. And it does do this in the context of some side effects. So we don't want to make it seem as if immunotherapy is a free ride of sorts. There are some side effects that can occur when patients have their immune systems activated. These largely represent the stimulation of inflammation to other parts of the body aside from the cancer. And although at the beginning some of these side effects could be quite severe, we as oncologists and immunotherapists now know quite well how to control these side effects. And through good communication between patients, caregivers, and the treating team, we now are very comfortable administering ipilimumab to our patients every day. So we were very gratified, of course, that there was an immunotherapy approved that provided benefit for patients with melanoma, but we also knew that the number of patients who were provided with this benefit was smaller than we wanted. And so another positive aspect of the approval of ipilimumab was really opening the doors to the development of other immunotherapies. The approval of this medicine, which can be simply administered in the outpatient setting, has spurred the development of numerous other immunotherapies by pharmaceutical companies in collaboration with academic investigators around the world, including many members of our Cancer Vaccine Collaborative. And another medicine that now is being looked at is another inhibitor of CTLA-4 called tremelimumab, which is part of the focus of the Cancer Vaccine Collaborative's partnership with MedImmune to look at a panel of different immune modulators in patients with a variety of cancers. And so we are now looking at tremelimumab, another CTLA-4 blocking antibody in a variety of diseases. It's being looked at alone and in combination with other immune modulators. And this family of what I'm calling other immune modulators now is typified by a family of medicines that blocks another molecular breaking pathway that also restrains T cells from achieving a state of full and persistent activation. And this molecule, or this family of molecules, is called the PD-1 pathway. And the PD-1 pathway is used by T cells, again, to prevent full and persistent activation. There are now 5 or 6 different medicines in clinical development that block either PD-1 or its partner molecules, which are found both on the cancer and on other immune cells. And several of these medicines have recently been presented. The data from clinical trials with several of these medicines has been presented at the most recent American Society for Clinical Oncology meeting several weeks ago, which have shown very impressive activity, not only in patients with melanoma and kidney cancer, but also in patients with common malignancies such as non-small cell lung cancer and gastrointestinal cancer. And I think this really is another very important step forward for the entire field of immunotherapy, that it is not just melanoma and kidney cancer that is responding to immune modulation, but more common malignancies, ones that oncologists see every day, such as non-small cell lung cancer, that is now being considered to be amenable to immune modulation. And so the PD-1 blocking antibodies, one of them is called nivolumab, another one lambrolizumab, and another antibody that is in development from Genentech, which blocks the PD-L1 pathway, is showing activity in a diverse panel of different cancers. The work that we presented at the American Society for Clinical Oncology meeting focused on a combination therapy of ipilimumab, the CTLA-4 blocking antibody, in combination with nivolumab, which is an antibody that blocks PD-1. And what we showed is that using this combination of immune modulating antibodies, we could cause regressions in 53% of patients treated with the doses that are going to move forward into a large phase 3 global clinical trial. And of those responding patients, of that 53%, all of them had a greater than 80% reduction in the amount of tumor that was present at baseline by the time of the first set of scans. So this is both a frequency of response and a depth of response. The amount of the decrease in the patient's baseline tumor burden was unlike anything that we had seen in prior immunotherapy trials. And so we are now beginning to build combinations of immunotherapies with other immunotherapies to try to disable several of the immune suppressive pathways that normally hold back T cells from optimally attacking cancer. But we are also looking at combinations of immunotherapy with other forms of anti-cancer therapy, such as radiation therapy. Our group and others have been studying the role of radiation as a way to sort of vaccinate from the inside, that when a tumor dies in response to radiation, the debris that's released and the change in the area around the tumor can actually cause a vaccination event. And if you supplement that with an immune modulator like ipilimumab or one of the PD-1 blocking drugs, you might lead to a very profound response. So we and others are performing clinical trials to test that strategy. We are looking at combinations of immune modulators with chemotherapies, properly timed, with so-called targeted therapies, which block key proliferative pathways that are active in a cancer, such as the BRAF inhibitors in melanoma or the ALK inhibitors in lung cancer. These are all rational things to begin to combine either concurrently or sequentially with immunotherapies to try to marry together the anti-proliferative effect of these targeted therapies with the ability of immunotherapy to induce durable responses to the cancer. So these are all areas of very active investigation, certainly within the CRI Ludwig Partnership Cancer Vaccine Collaborative and in other networks and clinical trials around the world. In addition, another area of immunotherapy which is being studied right now is a technique called adoptive T cell therapy, where immune cells are taken out of the patient's body. They are either expanded or they are genetically modified to make them more powerful or more specific to the cancer, and then reinfused into the patient. And there have been some very striking responses seen in melanoma of this adoptive T cell therapy at various centers around the country, the National Cancer Institute, as well as MD Anderson, the Fred Hutchinson, the Moffitt Cancer Center, all have generated some data in patients with melanoma. There are programs looking at something called CAR-modified T cells, where T cells are genetically engineered to express an antibody-like molecule on their surface. This is work that has been pioneered by Dr. Carl June at the University of Pennsylvania and Dr. Michelle Sadelain here at Memorial Sloan Kettering, who have shown very impressive responses in patients with both adult and pediatric leukemia. And so this type of cellular immunotherapy is also being looked at very carefully at a number of centers. Now, this is a type of treatment that is much more patient-specific. It does require special infrastructure, and we look forward to seeing how this type of genetically engineered or adoptive T cell therapy can be employed more widely across the country. And now that the pharmaceutical industry is also interested in this, we hope that this can be well applied. And actually, to again thank the sponsor of this program, Dendreon. Dendreon actually has shown that through their product Sipuleucel-T, which is a patient-specific therapy that tries to vaccinate against prostate cancer, that the manufacture and distribution of a patient-specific immunotherapy is possible. And so we look forward to seeing more efforts like that in the future. So those are the areas that I really wanted to touch on. So the history of immunotherapy, the beginnings with IL-2 and with vaccines, more recently with the development of immune modulating antibodies, and now also with adoptive T-cell therapy, really give great promise to the role of the immune system, which I think is finally being fulfilled in not just causing regression of cancer, but durable regression of cancer, because I think that is really the promise that immunotherapy has. We know that the immune system can remember the things that it sees for many years, and so we want that to be true not just for infectious diseases, but for cancer as well. So with all that being said, I'd like to try to answer the questions that have come in during my talk.
Sorry about that. I didn't have my microphone on.
I thought I was
Thanks, Dr. Wolchok, for that. Read my lips. No new cancers. Okay. Um, so, as you said, there's a lot of great excitement happening in immunotherapy right now. It seems like the field has really taken a major leap forward recently that has pushed us now out of some of the obscure cancers that responded very well relatively to immunotherapy, now into more mainstream cancers that affect larger populations of patients. So I think that's where we're going to see a lot of the real impact, and seeing more of these stories of remarkable successes happening for more patients. So we're certainly looking forward to that. So I do have a couple questions, and as I go through these, I would like to remind everyone that at the bottom of your screen is a Q&A field. If you have questions for Dr. Wolchok, feel free to submit them, and we'll go ahead and forward those to Dr. Wolchok. So our first question, Dr. Wolchok, is you mentioned inpatient treatment for immunotherapy, and so the question is, how is treatment with immunotherapy different as an experience for a cancer patient than we've seen or are familiar with other standard forms of treatment?
That's an excellent question, Brian. So some of the immunotherapy treatments need to be given in the hospital, such as high-dose interleukin-2. And that is similar to some chemotherapies, although most chemotherapy is now given in the outpatient setting. Some immunotherapy treatment is given intravenously, just like chemotherapy. And some of it is given in the doctor's office, just like chemotherapy is. So I think in terms of route of administration, many immunotherapies are similar. Some are shots under the skin. Some are intravenous infusions. Some are in the hospital. Some are out of the hospital. Some of the better known side effects that people imagine when they think of chemotherapy or any kind of anti-cancer treatment really are nausea, vomiting, and loss of hair. And these, in general, are less common with immunotherapy because the mechanism of action is different. It's not to say that there are no side effects from immunotherapy. It's just that the acute effects are quite different. Some patients will be tired after immunotherapy because of dysfunction of their thyroid gland or just their immune system working harder. And this is common to chemotherapy, but some of the side effects have to do with inflammation that is induced in various organs. So itching of the skin or diarrhea or irritation of the liver can happen with immunotherapy. Some of this can actually happen with chemotherapy as well, but I think most of the things that people normally associate with chemotherapy, the mouth sores and nausea, vomiting, and the loss of hair, are really uncommon with immunotherapy.
Okay, that's certainly good to hear, and I know that studies have been done, polls have been taken that show that people fear the treatment of cancer almost as much as they fear getting cancer itself. So certainly any improvements in a patient's experience in treating cancer is going to be good. Our next question is about the age of people who receive immunotherapy. It's known that certain chemotherapies, when given to pediatric patients, can cause lifelong debilitating effects, syndromes. of that sort. Do you see anything similar with immunotherapy, or are there what's happening in terms of immunotherapy for pediatric cancer patients, and what are the long-term impacts of
That's a
that?
great question, because we do know the long-term impact of the very important role of chemotherapy for pediatric malignancies. The simple answer is we don't know yet. The field is still pretty young, and there are not that many pediatric patients who've been treated with the most modern immunotherapy, so we simply don't know that yet. In terms of the other end of the spectrum, in terms of geriatric patients, there's sort of this assumption that the immune system may not be as potent a tool to use to treat cancer in patients of older age, because the immune system could slow down as people age. I'll just tell you that there are patients that we've treated in their 80s who've responded to immunotherapy for melanoma with some of the more recently developed anti-PD-1 medicines. So I think that while there may be some pre-existing thoughts about age and immunotherapy, we've certainly seen very young people respond in their early 20s or late teens, and we've also seen geriatric patients.
Well, that's really good news, because, as we know, cancer tends to affect older populations, given just the biology of the disease and all the things at play there. So it's definitely good to hear that some geriatric patients are responding well to immunotherapy. So here's a question that I want to throw out. You mentioned how these patients that you were treating with the combination therapies were having dramatic results that could be seen right away with the first scan. And this is something that we're used to seeing with chemotherapies, but it's not something that we're used to seeing with immunotherapy. Can you talk a little bit about the discussion that's been going on in the field about training people who are running clinical trials, training regulatory agencies that review clinical trials to understand the different mechanisms of immunotherapy, the time it takes for immunotherapy to kick in versus chemotherapy, and then how is what's happening now going to change that?
That's another really important point, and thank you for bringing up that question. That the usual thought process that patients and doctors have after giving chemotherapy or targeted therapy is that the first set of scans is extremely important in determining whether the treatment is working or not. And that's quite logical, that if you have a therapy that affects directly the division of cancer cells, then you just should see fewer of those cells and smaller tumors very quickly. With immunotherapy, it's a more indirect effect. You are not treating the cancer directly. You're treating the patient, and the patient is treating the cancer with the patient's immune system. And that may happen quickly if there are a significant number of T cells that are already there, but are being sort of held at bay by the immune suppressive forces of the cancer. But in some patients, that may take longer. And so what we have learned is that the most important thing to measure is how long people live, not necessarily how quickly their scans improve. We learned this very clearly in the development of ipilimumab, where the first scans were actually not very helpful in informing us about which patients would benefit and which patients would not. And it was, in fact, measuring how long people lived, and in fact sometimes doing a second set of scans to see who would benefit with just a little bit more time of observation. And we and other investigators have generated a new set of response criteria to hopefully inform the community of investigators as well as regulatory authorities about the importance of thinking about new response criteria that take into consideration the biology of the immune system rather than just the tumor itself.
Okay. Our next question from the audience is, when should a patient consider receiving an immunotherapy? As you said, a lot of these are still in the experimental phases, and that would require people to enter clinical trials. When is a good time for a patient to consider that? As soon as they hear that they have cancer? If they have a certain stage of cancer?
I think that's a really important point. For melanoma, since immunotherapy is such a major part of the approved portfolio of treatments for melanoma. I think that can be a discussion that happens as soon as someone presents with melanoma that can't be removed with surgery. And so there, I think it's quite clear that it's a discussion that should be had with the oncologist from the very beginning. In other diseases where immunotherapies are not part of the initial management, there needs to be a careful discussion between the physician and patient and family about what standard therapies have to offer and when immunotherapy could be incorporated into the discussion. And so, like I said, for melanoma, we have several immunotherapies that are part of the approved portfolio. In renal cancer, we have IL-2 approved for standard treatment of renal cell carcinoma. And in prostate cancer, we have sipuleucel T approved for the treatment of prostate cancer. So in those diseases, immunotherapy is part of the standard discussion. In other diseases, it becomes more of the conversation surrounding clinical trials.
Okay, thank you. We have another question. Some patients, when they learn they have cancer, biopsies are done, or in cases where it's possible, the tumors are removed as much as possible through surgical debulking or removal. Should patients have those tumor samples saved and preserved, or is this becoming part of the standard of care, or does it depend on where you are? And if so, why is it important to have those tissue samples preserved?
So it's always important to have tissue samples preserved. It's even more important now because so many of the therapies that are in use for a variety of cancers depend upon finding the presence or absence of specific gene mutations, because some of the non-immunologic therapies only work in patients who have specific gene mutations. So I think from the point of view of targeted therapy, the preservation and study of tissue at a genetic level is extremely important. It's part of the initial discussion that we now have with patients who have melanoma and lung cancer as well, because there are approved therapies, again, that depend upon a genetic analysis of the tumor. For immunotherapy, we do not yet have a predictive biomarker where a careful study of the tumor is going to absolutely dictate whether or not the patient can receive immunotherapy or not. It would be good, of course, to know that. But what we have learned from studying tumor specimens from patients at the time of diagnosis is the presence or absence or the amount of immune cells in that biopsy does provide some prognostic information about how well the patient may do in the long term. And that's another very important step forward that the field has made, that the role of the immune system from the very beginning is having a greater impact than we imagined.
So it's important to see what kind of activity is already happening, what kind of immune cell activity is already happening in and around the tumor. That can help you determine whether or not a patient is primed or ready or able to respond to immunotherapy, or whether or not it's better to pursue another course of treatment. So when patients come to Memorial and want to enroll in a clinical trial, how do they find out about that? Certainly at Cancer Research Institute, we have what's called a clinical trial finder service. Anyone can access it at cancerresearch.org/trial-finder, and it will help you find an immunotherapy for cancer treatment. But at Memorial and other cancer of course, Memorial is an exception. It's an outstanding cancer research institution, one of the leaders in this space. Is the experience at Memorial typical, or are people challenged based on where they are geographically to find the best care?
That's a great question, and thank you for the kind words about our institution. I think the internet has made this space a little bit easier to navigate. The National Institutes of Health, the National Cancer Institute, has a clinical trial navigator program called clinicaltrials.gov, which allows someone to search for a open clinical trial based upon various different criteria that you can use: your diagnosis, geography, type of treatment as well. So I think clinicaltrials.gov, in addition to the Cancer Research Institute's clinical trial finder, and also just talking to one's doctor. I think the world has just become a much smaller and more communicative place. And so I think there's a lot more dialogue between physicians, and that allows the patients to have access to more clinical trials.
Okay, so we're almost out of time. I do want one more question. A couple people have put this forward, and it's something that we get a lot at Cancer Research Institute. When you talk about the immune system and cancer, the general population might be ready to think about healthy living, eating well, getting lots of rest, getting lots of vitamin C, drinking your chicken soup, whatever it is that your mom told you to do to stay healthy. Is there any real foundation to that? And does that have anything to do with the kind of immunotherapy that we're talking about?
Those are great questions, and ones that we get asked all the time. And so we're still learning about this, about what are the role of sort of other complementary or integrative approaches to immune modulation. And various different groups are studying natural products for their potency in this space. My own advice to my patients is to follow what the American Heart Association tells you in terms of diet and a healthy living lifestyle. So get a moderate amount of exercise, as much as you can comfortably do. Stay as active as you can. Eat moderately. Don't necessarily go on any binge diets. Try to keep your nutritional needs satisfied. In terms of supplements or other natural remedies that one can buy, which may be touted as activators of the immune system, I think we just may not know enough in this area. And especially if one is on a clinical trial, one has to be very careful about the other things that one ingests, because unlike purified medicines, natural compounds may in fact have multiple thousands of different chemicals in them that are part of this natural product, and they may interfere or interact with the medicine that's being tested. And although taken for absolutely the right reasons, they may in fact cause more toxicity. We simply don't know, so we have to be careful. I tell patients I can't come and padlock your medicine cabinet at home, or police what you do in your own home, but I think it's just something that we have to recognize as being part of a big experiment that we're doing in a clinical trial, and the fewer variables that we have, the better off we are.
Yeah, so just about clinical trials, I want to say one thing, and I'm sure you have something else to add. This is what you do every day. The role of the patient in advancing scientific understanding for other people seems to be one that some patients really respond to. I know we've interviewed a number of patients who've received immunotherapies, and they've said that they really appreciate knowing that they're part of something larger than themselves. Whether or not the therapy helps them directly, they still derive some joy from knowing that they're going to be helping other people. Do you hear that sentiment a lot at the clinic?
Absolutely, we do. I mean, I think that people enter clinical trials for a variety of reasons. We wish that more people entered clinical trials. It's still a relative minority of cancer patients who participate in clinical research. And while there are many reasons for patients to, of course, choose standard treatment modalities, because they may work very well for patients with certain diagnoses, when the standard approved modalities are not providing benefit, then I think consideration for participation in clinical trials is very important. We are where we are today with new immunotherapies because of the courageous participation of patients in clinical trials. It's the only way that we're going to learn how new medicines work, and in fact, if they work. And there certainly are many serious considerations that patients have to make in terms of participating in a clinical trial. It is not an easy decision to make by any stretch of the imagination. All that we ask is that people consider this.
Well, with that, we're out of time. Unfortunately, we can't answer any more questions, but before I let you go, Dr. Wolchok, I just wanted to show you this picture, which we took in Times Square last week, standing in front of the NASDAQ board. There are a lot of faces in that crowd that I think you'll recognize. It looks like a lot of people from your own immunology lab.
Right, so this is
true?
basically the team that works in our laboratory, which is the Swim Across America / Ludwig Institute Collaborative Laboratory here at Memorial Sloan Kettering, as well as our human immune monitoring facility, which studies the very precious specimens from patients who are enrolled in clinical trials. And so this is a team of people that is singularly devoted to the task of using the immune system in new and better ways to try to conquer cancer. And we're very grateful to the Cancer Research Institute for, first of all, spearheading this Cancer Immunotherapy Awareness Month, but more importantly, for 50 plus years, for standing by the field, for educating our graduate students, our postdoctoral fellows, for sponsoring clinical trials, and for helping us get to where we are today. We're incredibly grateful to CRI for their real singular devotion to this now very popular field.
And of course, we are all, as our patients all around the world, grateful for the work that the scientists and clinicians are doing to bring more of these treatments to more patients sooner. Dr. Wolchok, thank you so much for being with us today. That was a wonderful presentation. Before we close, I'd like to just let folks know that next week, Tuesday, June 25th, is our 5th and final webinar in the series, Boosting Immune Defenses Against Cancer, with Philip D. Greenberg of the Fred Hutchinson Cancer Research Center in Seattle, Washington. You don't want to miss this one. Dr. Wolchok mentioned some new breakthroughs with adoptive transfer of T cells for immunotherapy. Dr. Greenberg is at the forefront of those efforts as well. I'd also like to draw everyone's attention to this wonderful promotion that our partner 1-800-Flowers.com is running during the month of June. You can buy from their Fight with White collection of flowers and gifts, with 10% of the proceeds coming back to the Cancer Research Institute to support our research. That's the link below. You can also access that from our website at cancerresearch.org/fightwithwhite. If you'd like to follow us on Facebook, Twitter, or our blog, these are the links for that. You can also find these on our website at cancerresearch.org. Once again, I'd like to thank our generous sponsor, Dendreon, for making this webinar series possible. And thank you all for attending. We hope that we'll see you next Tuesday. If you want to learn more, join the Fight with White at cancerresearch.org/fightwithwhite. That's all from Cancer Research Institute. Thanks again, Dr. Wolchok. Thank you all attendees.